Good morning, my name is Melina Moya and today I am going to talk about gene editing of CCR5 in autologous CD4 T cells of persons infected with HIV. Now let's talk about the definition. CCR5 is the major conceptor for human immunodeficiency virus, henchright. In this study was tested the safety and feasibility of inducing acquired genetic resistance to HIV infection to mimic the known inherited resistance displayed by persons with the CCR5 Delta 32 mutation. Let's see this video.
Has taken the lives of over 39 million people worldwide, despite our efforts to prevent, treat, and better understand it. But with 35 million people currently infected, what exactly is it, and are we close to a cure? To contract HIV, the virus must enter the bloodstream, and it's often transmitted from infected bodily fluids like blood, semen, vaginal fluids, or breast milk. Once inside the bloodstream, HIV targets a variety of cells, but most specifically the T helper cells, which are a type of white blood cell that play an essential role in our immune system and fighting infections. The outer envelope of HIV is covered in glycoproteins which mutate frequently, ultimately tricking the T cell receptors to not recognize the virus. Once attached to specific proteins on the T cell, it begins to fuse the membranes together and eventually enters the cell where it releases 2 viral RNA strands and 3 essential replication enzymes. Because HIV is a retrovirus, the RNA is transcribed into DNA, represented here by a zipper of 2 RNA strands transcribing into DNA. This DNA is then integrated into the host cell's genome. This makes the T cells treat the viral genes like their own, which causes them to make more copies of the virus. These then leave the host cell and mature, ultimately seeking more T cells. The virus is particularly difficult to treat because its mutation rate is so high. Overall, the replication process creates more than 10 billion new virions each day. During these initial stages of replication, called the latency period, a person may not show any major symptoms for up to 8 years. If not treated, the HIV eventually kills off the specific T cells it infects. When these T cells fall below 200 cells per cubic millimeter of blood, it becomes acquired immune deficiency syndrome, or AIDS. After progressing this far, the immune system becomes suppressed and is much more susceptible to cancers and opportunistic infections such as pneumonia. A person doesn't die from AIDS, they actually die from an illness that the body could not fend off. Nowadays, there is medicine that helps fight these opportunistic infections, like Daraprim, which was recently in the news when Martin Shkreli of Turing Pharmaceutical decided to raise the price from $13.50 to $750 per pill. There are also antiretroviral drugs that slow the virus down by blocking certain enzymes which are required for the virus to multiply. Similarly, those without HIV but at high risk of contracting the virus may take pre-exposure prophylaxis, or PrEP. This works similar to antiretroviral drugs by blocking the enzyme reverse transcriptase. Thankfully, there is hope for a cure. A small population of people are immune to the HIV virus because of a mutation linked to the T cells. In one case, an HIV-positive subject received a bone marrow transplant, meaning they were given new stem cells that generate different T cells, and within 20 months, there was no evidence of the virus in their bloodstream. Though this is very individualized medicine, it certainly opens up the possibilities of generating HIV-resistant cells. Combine this with other therapies and preventative measures like condoms, clean needle programs, and safe blood transfusions, and HIV/AIDS may one day be a thing of the past.
Now let's talk about the application in medicine. The study of gene editing of CCR5 in autologous CD4 T cells of persons infected with HIV, born from a preclinical test where CCR5 modified CD4 T cells expanded and functioned normally in response to matched genes, were protected from human immunodeficiency virus infection and reduced the HIV RNA levels in a humanized mouse model of HIV infection. Now I am going to present this video.
I think we're ready to really test your knowledge of what we learned in this lesson. There are a lot of really awesome concepts that we've learned really in all the lessons up to this point, and we can start building an entire framework around how to understand particular traits. Let's use an example that combines the concepts of mutation, selection, and allelic frequency. The CCR5 gene is located on human chromosome 3 and it produces a protein, the CCR5 protein, that you can see here in green, and it resides on the surface of T cells. T cells are immune cells that fight infection. Now, although T cells do a pretty good job at fighting infection, in fact, they do an amazing job, really, there's a virus out there that can really take them out. It's called HIV, or the human immunodeficiency virus. You might have heard of it before. The HIV virus binds really well to this protein called CCR5. And when it does, the whole complex gets internalized. And this is how HIV gets inside T cells. It hijacks this protein and uses it to get inside the T cell, infect it, and then infect the entire immune system. So if you have a normally functioning CCR5 allele, then you're at risk for HIV infection. However, there's an allele of CCR5 that creates a very dysfunctional version of the protein, rendering it pretty much non-functional, and it can't even get to the cell surface of T cells. Without that CCR5 protein, the HIV simply can't get in the T cells. The T cells are thus resistant to HIV infection. So here's your first question using this example. Is the mutation for the dysfunctional protein that makes the T cell HIV resistant more likely in the coding or non-coding DNA of the genome? I want you to check one of these boxes and then explain why you think that's the answer in this text box here. I want you to save your answer because guess what? You're going to need it.
Let's talk about the pros and cons. About pros, CCR5 disruption seem likely to increase the survival of CD4 T cells. Persons who are heterogeneous for CCR5 delta 32 and who have HIV infection have a slower progression to the acquired immunodeficiency syndrome. And of course, the blood levels of HIV DNA decreased in most patients. About the cons, a serious adverse event occurred in a single patient, with fever, chills, joint pain, and back pain developed in the patient and precipitate a visit to the emergency department within 22 hours after infusion of the study drug. Let's see this video.
So there's this thing called CCR5 gene editing trials. There's been some discussion about this. Do you know if that's happening in Australia? Is that one of the ones you've been talking about?
Gene therapy is another approach, and what CCR5 gene editing does is it basically makes someone's cells resistant to HIV. So HIV needs this protein called CCR5 to get inside a cell. It's like it uses that protein. It's like a lock and key combination. You've got to have CCR5 there and then the virus can slip inside. But we can now do gene therapy, which effectively means chopping out the gene with these sophisticated tiny micro gene scissors that can get rid of the CCR5 protein. That's what gene therapy is. It gets rid of the gene that you don't want anymore. And so that we can do that. So you can edit out CCR5. Basically how those studies have been done, and at the moment they're only being done in the US. A patient with HIV on treatment gives a large amount of their blood. Those cells are edited by these gene scissors and then they're put back into patients, these now CCR5 negative cells. And then they're resistant to HIV. At the moment, those studies look promising for a range of reasons. First of all, it's safe. There's no adverse effects. Those modified cells survive after you put them back into someone, but they're still at very low levels, around 6%, and they probably need to get to much higher levels to be effective. But a lot of interest in that. I should say one thing about gene therapy is it's expensive and it's very complicated.
Now let's talk about the issue applied in Ecuador. In Ecuador, an average of 10 people is infected with HIV daily. Today, the life expectancy of people living with HIV, once contracted, is highly dependent on timely and continued immunization, antiretroviral treatment, and adherence to treatment. Because of that, it is important to know this new study, which pretend to lower the blood level of HIV in most patients. Now, to conclude, let's talk about the conclusion of this study. CCR5 modified autologous CD4 T cell infusions are safe within the limits of this study. This study supports the feasibility of targeted genome editing to introduce a disease-resistant allele. Thank you.